Okay, you guys. On Friday, we were talking about enzymes and exactly how enzymes work. The enzymes have that special spot on them called an active site where the substrate binds. And the enzymes are very specific, so the specific shape of the enzyme works with a specific substrate and therefore catalyzes or speeds up a reaction by one of two different ways. So remember that some enzymes work by breaking things down. So this was the example with catabolism, that here's the active site and it breaks it down and it's very specific because of the shape. Or it can build things, and so in this case here it was anabolism, where it is putting together two molecules and building a larger molecule there. So those are two examples of the enzyme working. And so then this last thing with the induced fit. So if you recall that enzymes have this special shape. So this is the three-dimensional, this is a chain of amino acids. So all these little balls in here are the atoms that make up the chain of amino acids here. And it has this three-dimensional shape. So remember with proteins they have the coils and the pleats and then they fold on one another and the R groups interact. So you have this three-dimensional shape. And the active site is the important part here that bind the substrate can. Remember, they're randomly moving around. The substrate binds into the active site. And then the enzyme just changes shape just slightly. It kind of goes and kind of hugs the substrate a little bit. Or another analogy is a clasping hand shape. The hands come together and then form together. And so that's what is happening here. And so the enzyme, the active site just kind of goes around the substrate a little bit. And then remember, it can produce the product by either if they're building something, two substrates together, they orient them so that the parts that bond together come together, and therefore they can bond easier. Or they can, if a molecule is a bigger molecule, it's going to break it down. When it does the induced fit, it can put stress on the bonds. So it puts stress on the bonds, which makes the bonds break easier. And so either way, it reduces the enzyme, speeds up the chemical reaction by reducing the what? What we talked about on Friday? That some reactions need an initial energy. Scott?
It reduces the activation.
It reduces the activation energy, and so therefore if the amount of kind of startup energy needed for the reaction to occur goes down, the reaction can occur quicker because you need less input of energy from that. All right, and so that's the enzyme, and that's where we left off with on Friday. So then your homework kind of went over and introduced you to things that affect the rate of reaction for an enzyme. And so temperature and pH are one of those things. So an enzyme's activity can be affected by temperature, pH, and chemicals. And so basically these three things, how they're affected is that most of the time it changes the shape of the enzyme, which then changes the shape of the active site, which then the enzyme can't bind with its substrate if it's changed shape, and so therefore can alter its activity. Let's just talk a minute about this optimal temperature for the two enzymes here. So this is the typical human enzyme. The optimum temperature is at 37 degrees. Notice here it's at the peak here. This is the rate of reaction, so at 37 degrees is the highest rate. And then as we go either to the left where it gets colder or to the right, the rate changes and the rate slows down. But they don't slow down at the same rate. If you look here, it takes a lot longer for it to come down to zero and no rate of reaction as it gets colder. It happens much quicker as the temperature increases. Can somebody tell me why it looks different from cold versus hot? Why does it take it so much longer for that rate to slow down than it does when it's hot, when it's cold? Have an idea? Megan?
Does it have to do with the heat increasing the kinetic energy in the protein of the enzyme, and so it disrupts it quicker because of the change in kinetic energy?
Now with the heat, let's talk about that heat. Heat increases the kinetic energy. So remember, this enzyme is moving around. So not only is it moving around, the cell at a quicker rate when it's hot, but also the atoms that it's making up that enzyme are moving. And so remember that what causes that enzyme to have that three-dimensional shape in the first place is the interaction between the R groups. And so what happens is, as this is moving around faster, the R groups, that interaction disrupts, and then the protein unravels. If it unravels, then it doesn't have a shape anymore, right? And the active site certainly isn't the right shape, and therefore there's no rate of reaction very quickly. And so that's what's happening here. So what do we call that when the enzyme loses its shape? Denatured, right? So when it gets hot for this, the enzyme is denatured. So that's why it breaks, that's why it drops very quickly. When it gets cold, is the enzyme denatured? No, the enzyme is not denatured. So what's happening here in the cold as it gets colder to cause that rate of reaction to go slower over time? Jacob?
Slowing of the particle or the atoms and the enzyme. Yeah.
Yeah, the slowing of them. So remember, for a reaction to occur, the enzyme has to meet up with the substrate. So these guys don't eyeball each other and beeline it for each other to bond. So they're constantly just randomly moving around. And so when the temperature slows, they move slower and slower and slower and slower. And so the chance that they ever move and actually come together for a reaction to occur is very, very slim. And so that happens as it gets colder. They move slower and slower and slower. So this enzyme and this substrate have a less and less chance of getting together, and so therefore the reaction goes down and down and down. So this is just because this part here is that the enzyme is moving slower. And doesn't come into contact with the substrate as often. And doesn't come, I'll write this down because when you look at this later, it'll make a little bit more sense. It doesn't come into contact with substrate as often, hence making the reaction slower. This is the one, temperature is the one thing that makes it's different for cold versus hot. So our typical human enzyme, roughly about 37 degrees Celsius, that's about 98.6 degrees Fahrenheit. That's why our body works very hard to maintain that constant internal temperature. Why? Because our enzymes work best at that temperature. So we get a little bit too high, so high fever, bad news, all right? If it gets too high, your enzymes can denature, and then chemical reactions that keep you alive can't happen, and it can be life-threatening. All right, so too high of a temperature, not good, and this is also what happens if you were to, go out in the cold. Your body's going to do certain things to help keep your temperature at 98.6 degrees, like shiver and so on and so forth. But if it's too cold and your body temperature keeps lowering, your enzymes are not going to come together with those substrates. It's going to slow those rates of reaction down, and we can freeze, all right, and die. All right, as a result of that. All right, this little guy here is the heat-tolerant bacteria. This just shows you that while this is true for the typical human enzyme, this is not the case for every living thing on Earth. So there are bacteria that live in very hot environments. They live in hot springs and so on where the temperature is very hot. And we would not be able to survive because that is out of our optimum temperature range. But the bacteria are able to survive, and that's because they have different enzymes, and those enzymes have a higher optimal temperature. So you can see here about 77 degrees Celsius, much higher than ours. That's their optimum. But notice the shape of the curve for these guys. The curve is the same shape for the heat-tolerant bacteria as it is for the human enzyme. It's just the optimum is at a different temperature there. So therefore, when it gets, for these bacteria, if it gets much lower than 77, it slows down the rate of reaction, just like it does for us. And if it gets much higher than 77, it drops down very quickly, just like it does for us. So they're adapted to a different environment there. Okay, so that is temperature. Then pH. pH, this is going with 2 examples in humans in our digestive system. The stomach pepsin enzyme and the intestinal enzyme trypsin. And so this is a specific example, a unique example in us. Most of our enzymes, the optimal pH is not so acidic. All right, so what is this, about 1.9 or so? It's just slightly to the left of 2. And so this is unique for this particular organ, where the enzyme pepsin works best in this acidic environment. If you get too acidic, so to the left is acidic, or too basic, it goes down pretty quickly. So it works, doesn't mean that it doesn't work at 1.5. It does, it just slows down until eventually it doesn't work, all right, and vice versa. So what happens with your stomach, let me just, kind of sidetrack here with your stomach. I forgot to charge this, so my little thing doesn't, I have to move it so it shows the thing. Your stomach, if I were to look at the lining, so this is the thickness of your stomach, this is where your food is. So there is, I'm not a good artist here, but there's your esophagus, all right? Here's your mouth. Picture this. All right, you have to use your imagination a lot. All right, so here's your mouth, here's your esophagus, and there's your food. So you chew, all right, it goes down your esophagus, just leads to your stomach. There's a little muscle that opens and closes. So it opens and allows food to go in, and it closes it so your food doesn't go back up your esophagus. So here's your food. The lining of your stomach has different types of cells. They have cells that produce enzymes and secrete the enzymes. This would be through the endomembrane system, where they make the enzymes, and the rough-ER, then it goes to the transport vesicle, to the Golgi apparatus, and then it goes from the Golgi apparatus to the cell membrane and secretes these enzymes. The enzymes are actually what digests your food, but the enzyme, like pepsin, works best at a pH of 2. So guess what? We have to have an acidic environment in our stomach. So you have other cells that produce acid. So other cells that secrete acid into your stomach. Now, if you were to dump acid on your hand, is that a good thing? No. It's not a good thing. So this is the reason for our third type of cell. You have another type of cell that secretes mucus. And this mucus actually goes all the way around here. It's like a liner on your stomach, and it protects your stomach from the acid. So the purpose of the acid, in this case, is really not to digest your food. The purpose of the acid is to create the right environment for your enzymes that work best in an acidic environment. Works best in the acidic environment, and those enzymes will be in the right shape and will be able to digest your food. Okay, so that's a unique situation. Most of our enzymes in our body do not work best at such a low pH, but your stomach is unique in that particular case. So then, as we go to the small intestine, so the small intestine this is where your food goes next, all right? So there's a little muscle here that allows the food to leave then your stomach and enter into your small intestine. And this is where further digestion happens. So there are other enzymes like trypsin. So this works best at right around a pH of 2. These guys work best at what? About a pH of 8. So now you have this, they call it chyme. It's like part digested food mixed with the enzymes, mixed with the acid, all right? And now it's it goes out of your stomach into your small intestine, and then look at, your small intestine doesn't have the mucus there, and so what do we have to do real quickly to the pH? We have to make it more basic, right? And so therefore there's a solution that is dumped into your small intestine that is a basic solution to bring that pH back up, because our enzymes that are in our small intestine that are going to further digest our food work best in a pH of 8. All right, and plus we don't want to damage the small intestine that doesn't have those special cells to produce the mucus to protect it. And so, so therefore we change that pH, and so now the enzymes in your intestine, this is not the only enzyme, but the intestines are the enzymes in your intestines will be able to work at their optimal pH and continue your digestion of food. All right, and so those are two examples of two enzymes within the same organism that have different optimal pHs. And this is why, like the food here, these muscles that open and close, if your food gets back up into your esophagus, that's what they call acid reflux. You guys have heard of that before? All right, and so it's when that muscle opens up when it's not supposed to, and some of that food gets in there, and the food has the acid mixed in with it because we're digesting it, so your enzymes can digest it, and your esophagus does not have the mucus to protect it, so therefore you get this burning sensation, or sometimes that's why they used to call it heartburn. Now they call it acid reflux, but they call it heartburn as well. Because why? Because it feels like where your heart is, all right, where you feel it, but it has nothing to do with it. All right, and so that's that. All right, so that's your pH, and then certain chemicals can also interfere with the R group's binding, and therefore certain chemicals can also cause the enzyme to change shape. So all of those things can affect how our enzymes work. Some other things that can affect how our enzymes work are called cofactors. Co in front of something means they're going to work together, so it's going to work together with an enzyme. And some enzymes need other molecules to work properly. So a cofactor are non-protein enzyme helpers, such as minerals like zinc and iron and copper. So we have some enzymes that don't work properly unless you have a proper amount of these minerals in our diet. So zinc, and so there are certain enzymes that will not work properly and not have the right shape unless zinc is present or iron is present, and so on. There are special kinds of cofactors called coenzymes. These are organic. Move this so we can see it better. These guys are organic cofactors. In chemistry, what does organic mean? The molecule contains something. It's not natural. A lot of people want to say, oh, it's natural, like when we think about going and buying organic pepper at the grocery store, all right? That would be all natural, no fertilizers, pesticides used, and so on. In chemistry, organic means what, Colby?
It's carbon.
Yeah, it has carbon. Or contains carbon. So a lot of vitamins are organic cofactors. So the coenzyme is just a special name for a type of cofactor. So cofactor is just something that is needed by an enzyme to work properly, and a coenzyme is if that thing is organic. See, zinc is not organic, all right? So it does not have carbon in it. And so a lot of vitamins are. So there might be some enzymes that won't work properly unless you have enough vitamin B12, let's say, okay? And so therefore having these cofactors kind of regulate and if the enzymes are going to work properly or not. So these guys kind of activate the enzymes and work together with the enzyme to make it work. There are some things that inhibit enzymes from working. And so there are two different types, two different categories of inhibitors. Competitive inhibitor binds to the active site of an enzyme and competes with the substrate. So the inhibitor and the substrate are competing for what? What are they competing for? The active site. So let me draw up here. So this is your enzyme. So this enzyme has a particular shape. This is the active site. That's important. So what happens is you can have, let's say, a molecule that has this shape that can bind to the active site. Or you could have a molecule of shape like this that can also bind with that active site. Two different molecules that can compete for that active site. So let's say this is the substrate. And this is the competitive inhibitor. They can both go to the active site, and depending upon which one, depending upon which one gets there first, the reaction may or may not occur. If the competitive inhibitor, if these guys are bouncing around, and the competitive inhibitor kind of gets to the enzyme first, then is the reaction going to occur? No. If the substrate gets there first, then the reaction will occur. And so therefore, obviously, if you have no inhibitors, the reaction rate will be the highest. And then as you introduce and have inhibitors, it can stop the enzyme from working by physically blocking the substrate. So this does what? Inhibitors decreases the rate of reaction. All right. So sometimes we can combat that by, let's say, if you increase your substrate concentration compared to an inhibitor, what's that going to do to the rate of reaction? Oh, yeah. You weren't raising your hand.
No.
It's going to increase the rate of reaction compared to the competitive inhibitor. So we can combat that, but in general, competitive inhibitors decrease the rate of reaction. The other type of inhibitor is called a non-competitive inhibitor. It binds to another part of the enzyme. The enzyme changes shape, making the active site non-functional. So let me draw what this looks like here. So here is, move this over here. So here is your enzyme, here's your active site, and then I'm going to draw another site separate away from the active site here. So this is still your active site of your enzyme. And this is still the shape of your substrate. So here's your substrate, can fit into there, so that's your substrate. But a non-competitive inhibitor is going to be a different shape and therefore bind to a different part of the enzyme. So I'm going to put it kind of diamond shaped here. So it can bind to that part. So this is non-competitive inhibitor. Now, when it actually goes and binds to that, how that's going to change the shape is that, I'm going to draw it down here. So when this happens, this enzyme, so you still have the other site here, changes the shape, and the important part that gets changed is the active site. So now we see that here's your non-competitive inhibitor bound to that site still, but the enzyme has changed shape, and the important part, the active site, has changed shape. So now our substrate here cannot bind. And so therefore, we also lower the rate of reaction in that case. And so both of these, competitive and non-competitive, both lower the rate of reaction, just in different ways. Okay, so this one's not changing the shape, but it gets there first, and this one's actually changing the shape. But in order to, the non-competitive would have to get to the enzyme first before the substrate. If the non-competitive inhibitor didn't get there, the substrate got there first, it would still, the rate of the reaction would still occur, all right? So those are two different inhibitors. A lot of poisons work as inhibitors, and that's why they're poisonous. So like different chemicals that you take into your body can actually inhibit enzymes, and if they're important enzymes that catalyze important reactions, it can cause death, okay? Or adverse reactions.
Why does that change of shape occur with the active site?
Yeah, because remember that the enzyme is this chain of amino acids and has a specific shape. So what happens is when this is really simplistic here, the drawing here, but when that inhibitor binds to it, it interferes with the R groups and kind of causes it to unravel a little bit so that they're binding with the inhibitor instead of each other, and therefore changes the shape slightly.
Okay.
Okay?
I'm just curious, so if the substrate came into the enzyme first, could the non-competitive inhibitor still come in and change the state, so the substrate would have to like leave?
No, so if the substrate got there first, it would be converted to product, but the product leaves, and then it could bind to it next, and therefore inhibit the next substrate from, alright, does that make sense? Okay, yeah, so it's not together, so it's kind of whoever gets there first wins, alright, whether or not the reaction is going to occur or not. Any other questions? Yeah.
Would a non-competitive inhibitor ever change the function of an enzyme so that it still works just for something else?
That is possible, yes. It's possible. Off the top of my head, I can't think of an example of that, but I would say that that's probably possible. Yeah, absolutely. Any other questions? Yeah.
Could inhibitor bind to the substrate instead?
Could an inhibitor bind? Then it wouldn't be called an inhibitor. There are molecules that you could take into your body that could bind and react with the substrate, preventing that reaction from occurring, absolutely. But it wouldn't be called an inhibitor because it's not really inhibiting the enzyme from working. Does that make sense? Okay.
Yeah, it's more indirectly.
Yes, yes. It's stopping the reaction from occurring, but not by inhibiting the enzyme. All right. Other questions? Good questions. All righty. So let's go. You don't have these particular pictures, but you have another one here. But just look at this one real quickly here. So if we look at it, it says competitive inhibition here. What is in this picture here, what's the substrate? Think about what color the substrate is. What color is the substrate, Michael?
The red thing with the yellow, blue, and
Yeah, the red thing, what do we call it? Is it purple to you? Purple or blue?
It's purple.
It's purple, alright. Purple, the purple thing, that is what in this picture? Your competitive inhibitor. So I want to point out here with this, that the competitive inhibitor, so here's your active site. Alright, the active site is here. The substrate has a complementary shape, but the competitive inhibitor, notice here, it doesn't have to have the exact same shape, alright, as the substrate. It just has to have a somewhat complementary shape that will actually bind in that active site. And so it doesn't have to have the exact same shape there. And so that's your competitive inhibitor, and here's your non-competitive, just to show you a little bit different picture than my picture up here. Here's that site away from the active site. If that non-competitive inhibitor gets there first, it changes the shape of that active site, now not allowing the substrate in. All right, that substrate reminds me of a little bird. Head of a bird. All right, so this is what you have in your sheet. So just compares and contrasts normal enzyme, what it looks like, normal shape, and the substrate that fits into that, competitive versus non-competitive. Both of these stopping the reaction from happening. All right, so let's look at how we regulate regulate enzyme activity. So you have literally thousands, tens of thousands of enzymes in your cells and in your body that are catalyzing and speeding up reactions, thousands of reactions at any given second, and to keep us alive. Let me ask you this, what makes enzymes? What controls the making of enzymes? How does a cell make enzymes? Where's the directions at? Megan? Yeah, with the nucleus and the DNA. The DNA, your genes and your DNA contain instructions to make all your proteins, and enzymes are proteins. All right, so we make the enzymes. Now that the enzymes are in the cells, let me ask you this. Out of your tens of thousands of enzymes that we have the ability to make in our cells, do we want every single enzyme working at every single second of every single day, do you think? No, and so therefore there are certain times, like when I had the stomach up here as an example, those cells that are releasing enzymes to digest your food, it would be very wasteful for them to continue making enzymes and putting enzymes in your stomach when you haven't eaten in a day. All right, so there's no food to digest in your stomach. It'd be a very wasteful process. So therefore they only make them when food enters the stomach. And so therefore we have to have a way to regulate enzyme activity for enzymes to work sometimes and not at other times so that we're not wasteful. So that's what we're going to look at. To regulate metabolic pathways, the cell switches on and off the genes that encode specific enzymes. So we can actually turn genes on and say, oh, we're going to make the enzymes now. Oh, we're not going to make the enzymes now. All right, I'm introducing that to you now. When we get to genetics, we'll talk you exactly how that happens. And we call the enzymes that we do this with, we call them allosteric regulation. A protein's function at one site is affected by binding of a regulatory molecule to a separate site. What's called the allosteric site. So I'm going to introduce these regulatory molecules. So this is new. So enzymes are going to work depending upon, or not work, depending upon if another molecule is present or not. So those regulatory molecules could be an activator, just like it sounds, stabilizes the active site, it activates the enzyme by making sure it's in the right shape, and so that the enzyme works, and stabilizes that shape. On the flip side, an inhibitor stabilizes the inactive form so that the enzyme isn't in the right shape and therefore can't work. So it's going to inhibit the enzyme from working by changing its shape, and so the active site is not the right. And then we're going to round out with cooperativity. One substrate triggers the shape change in another active site, which is going to increase catalytic activity. Catalytic activity, what in the world does that mean? Catalytic. Sounds a lot like catalyst, right? What does a catalyst do? Speeds up a chemical reaction, right? And the catalyst in living cells is called enzymes, right? So when it increases catalytic activity, it increases enzyme activity in this case here. All right, so that's what it's talking about. So making the enzyme work better. So you have this picture here, and I'm going to write on here with you. Get a blank copy out of here and go over it with you. I want to give you an example of how this works. So I'm going to relate what we talked about on Friday. Friday we talked about enzymes, or actually it was Thursday we talked about what we're going to use this too. We talked about enzymes catalyzing certain reactions. So I'm going to draw on this with you. But before I do, let's review the reaction that we're going to talk about. And that is the use of ATP. So ATP is our main energy molecule in our cells, right? How do we get energy from ATP? Anna, what do we do?
It's when the last positive or something.
That's right, when the last phosphate breaks off and it becomes ADP. I forgot to do. So, so that's hydrolysis, so we add ATP in water, and it breaks down into, as Hannah said, ADP, and that when that phosphate breaks off, if it's not attached to anything, it's called an inorganic phosphate. This reaction, is it exergonic or endergonic? Does it release energy or need energy? So this releases energy. So this releases energy to do work for the cell. So this is our main cellular energy, but you can see here that if we could eventually run out of ATP. So therefore, to regenerate ATP, we put the phosphate back onto ADP, and in that process, we make ATP. In this process, is this endergonic or exergonic? This is endergonic. It needs an input of energy. My question now is, where does that energy come from? It's the main thing that provides energy to make our ATP, to put that phosphate back on. Madeline?
Is it the energy that's released from the breakdown?
Actually, it's not. It's a good hypothesis, though. But the energy, this energy usually does work for the cell, and this energy usually comes from the breakdown of another molecule. None? Sugars. All right, so glucose being the main one. So breakdown of sugars or glucose. That process is called cellular respiration. So we break down glucose and energy is released. That energy is used to put the phosphate back onto ATP. So this I wanted to review because I'm going to use this as an example of how this allosteric enzyme works. So let's look at this. So I'll color code here a little bit. When we look at this, let's not look at the right-hand side right now. Let's just look at this picture right here. So this is your enzyme. This allosteric enzyme has a different shape than the enzyme I drew up on the board. This allosteric enzyme has 4 subunits. So you can see here 1, 2, 3, 4 subunits. Let me move this here so we can make sure. 4 subunits, and each subunit has an active site. So how many active sites does this enzyme have? 4, right? So we look here, I'm going to color code them just so we can see. Here's an active site, here's an active site, here's an active site, and here's an active site. So active site, 1 of 4. Then I want to point out that if you look at the space in between the subunits, right here, there's another site. These guys here, they call this site the regulatory site, and so there's 4 of those in this one. This is otherwise called, I'm going to put here, or allosteric site. So there's a different site away from the active site, and that's the active form of this enzyme, meaning that this is the right shape of the active sites to make this enzyme work. So this is the active one. Now, not looking at the right-hand side, if we just look at the bottom, it says oscillation, and you notice that there are arrows that go back and forth here. So this enzyme can oscillate or be changed between the active form and the inactive form. So here's your inactive form. If you notice here, the inactive form, what has happened to the active sites? Yeah, it says non-functional. Why are they non-functional? They've changed the shape, right? So these guys have changed shape, and therefore the substrate cannot bind to it, and therefore it's in the inactive form. So what we're going to talk about is, remember we said that we don't want enzymes to work all the time. So therefore, there are certain situations where you want the enzyme to work and it to be in the active form, and certain situations where you might not want the enzyme to work anymore, and so therefore we convert it to the inactive form. So that's what it means by oscillating back and forth, depending upon if that reaction needs to occur in the cell. So I'm going to give you a specific example of this using ATP and glucose as an example. Okay. So I'm gonna write some things along the sides, and everything to give you, a clear picture as far as when their cell would use this. So let's say this is an enzyme. That helps break down glucose. So we wanted to help break down glucose. So if we look here at the enzyme in the active form versus the inactive form, we would want it to be in the active form and help break down glucose when we need ATP, right? And then you would want it in, when would you want it in the non-active form? In what situation would you not want to break down glucose? Colby?
I mean, if you're not doing work that's necessary, so like sleeping maybe?
Yeah, absolutely. So there are certain times where you use more ATP than others. So if you're out running a marathon, you probably want to be breaking down some glucose to make some ATP because you're going to need it. But afterwards, when you go lay on the couch and fall asleep, you don't need as much ATP as that you did when you were running the marathon, right? And so therefore, that would be an example of when you might not want to slow down the breakdown of glucose and making ATP because you're not using it as often. So that's what we're going to talk about, how that works, because that's exactly what your cells do. So let's write this here. If ATP production lags behind its use, before I finish that sentence here, let's look at that chart on the thing on the board. ATP production lags behind its use. What's happening more, the reaction on the left or the reaction on the right? Meaning, is ATP being made or is ATP being broken down more? ATP production lags behind its use. So ATP production, that's this, lags behind its use. That means this side is happening more. So you're using ATP a lot, but we're not regenerating it at a fast enough rate. So we want to make this happen faster, all right? So what happens is if you're using ATP a lot and your cells aren't regenerating it, do you see where you would get a buildup of ADP? Because you keep breaking down ATP and not reusing it to make more ATP. So then what happens is, the rest of that sentence is that ADP accumulates. We get a buildup of ADP. So if ADP accumulates, and that means we're breaking down a lot of it, of ATP, we need to produce more ATP. And what's going to help produce ATP? What are we going to want to do to produce more ATP? We're going to want to increase what? Cellular respiration. We want to increase the breakdown of glucose, right? Because that's going to, the breakdown of glucose is what provides the energy there. So therefore, we need to break down more glucose. To provide energy to make more ATP. So we want more cellular respiration to happen. So if this is an enzyme right here that helps break down glucose to make ATP, in this situation, do we want this enzyme to work or not? This is an enzyme that helps break down glucose. Do we want that to happen? We do. So we need this enzyme to work. We need it to be in the active form. So in this particular similar situation, we just said that when you're using up ATP at a fast rate, you get an accumulation of ADP. So let's look at this. To stabilize in the active form, we have this little activator. Let me use a different color here, pink here. So here's your activator. In this case here, ADP acts as an activator. Because what happens is you're using up a lot of ATP and not making it, so you get an accumulation of this ADP. This ADP, notice here in this picture, here binds to the regulatory site. So I'm going to put here that ADP, when it binds to this site, ADP stabilizes. Just so we can actually see. ADP stabilizes the enzyme. So glucose can be broken down to release energy to make ATP. And what I mean by stabilizing, so in our cells, as you use up ATP and you get this ADP accumulating and you're not using it to make any more ATP, all that accumulated ADP binds to that regulatory site, the allosteric site, and makes it so that the active sites stay in the right shape. So now your active sites are stabilized, and that means that it can't change form. Stabilized active form means that it stays in the active form, and this enzyme is working, and therefore it's going to break down glucose and start using, releasing energy to do that left part of the equation up there, that left part of that cycle there of making ATP. All right. So one other thing that I'm going to add at the bottom of this, the last thing over here, is that ADP acts as an activator. Okay, so this would be like when you're running your marathon. You're using up ATP, maybe, so you started running, and all of a sudden you've changed the quantity of ATP that you need. And so you're using up your ATP, and we need to change how much the rate of cellular respiration. So then, as we talked about earlier, as Colby said, what if you go home and you go to sleep? Now all of a sudden you're using less ATP. If you're using less ATP, you don't need to make it as much, right? And so let's do the last little bit here and talk about the second picture here. So if ATP is being made faster than it's being used, Oh, come on, day morning. So what are you going to have? You're going to have too much of, if you make ATP faster than you're being used, instead of having an ADP accumulate, what are you going to have? You're going to have too much ATP, all right? So ATP will accumulate because you're not going to be using it. All right, this girl's not the same.
All those factors, like some of it being clear, like, those factors can cooperate with
I know, I got to it first. How what happened here? Yes. Right. So remember, you guys, that packet that I gave you on Thursday is due tomorrow, okay? And we'll wrap this up tomorrow.